moving directly to our next speaker, Rob Finn, who is a team leader at EMBL-EBI Center. He's a world leader in microbiome dataset sharing, such as the Magnified database he's leading, which enables all of us to analyze, discover, and compare microbiome data. And I think you'll see examples very shortly. Rob started in microbiology, but after frustrations following 48 hours of continuous protein purification, he came to the conclusion that the wet lab was not where his strengths were. Happily, he has many strengths. And so after his PhD, he changed his career path to bioinformatics. And Rob, please tell us about all the interesting things you found in that new world.
Thank you. Brilliant. Okay, so thank you for that introduction. And as Ron mentioned, I work at European Bioinformatics Institute as part of EMBL, and I run a resource called Magnify. And what I'm going to do is talk to you not so much about the science, although there will be some element of science, but also about how we run these resources. So one of EMBL's core missions is not only to do research, but also to deliver science resources for the community. But what I really want to do is start to try and shed some light on how these huge data sets, these metagenomic data sets, can actually help tell us about how we can heal the planet. So your quick 101 on metagenomics, so for those who may be not so familiar, is on this slide. So metagenomics is really the study of the entire microbial community that's living in a particular ecosystem. Now, on this slide, on the left-hand slide, I've got the big three that I always refer to in terms of microbiomes that have been particularly studied. So there's been marine microbiomes, soil microbiomes. Diane's already mentioned a bit about soil microbiomes, and then the human gut microbiome. But since the field started, when we first started looking into those microbiomes, the techniques and sequencing isn't as cheap as it was, as it is now. And so often people could only look at who was there. They would use a marker gene, the so-called metabarcoding, and actually just use that gene to work out who was present within an ecosystem. And typically that marker gene would be something like the ribosomal RNA, the SSU, but there it would be restricted to just looking at the bacterial component. Since then, sequencing has got much cheaper and the interest in the field has increased. And so now with diminished sequencing cost, we can actually look at the whole community. So we can look at the viral component and the single cell eukaryotic component. And now we tend to look at the whole sum of the DNA, the so-called metagenomic part. That's where you're looking at the total DNA. You can also look at actually what part is metabolically active by looking at the metatranscriptome, although that's generally a bit harder because extracting the RNA is more difficult than extracting the sum of the DNA. But since the field has expanded and we can start having not only who's there, but what those microbes may be doing, this area, the field has diversified in different applications. So there, on the left-hand side, again, there's the flasks that indicates this notion of green chemistry. Can we find enzymes that can carry out jobs that previously chemical synthesis would perform? Can we understand biofuel production that microbes can be involved in? So can we go into silage heaps and understand those microbes? And the oil industry is also very interested in this. We've heard about how microbes can clean up oil spills, but their microbes are very good at actually going into oil fields and polluting oil fields with hydrogen sulfide, so-called oil field souring, and they also form biofilms on the inside of pipes that cause corrosion. All of this leads to a huge different diversity of microbes, the complexity of the communities, and how we can study them. And what I do is produce a resource that tries to capture all of this data that's coming in across the community and actually provide a free-to-use resource that not only captures this data, archives it with the associated metadata, assembles this data where appropriate, and then provides the analysis. And this is a free-to-use service. So what we do is the approaches that we use can be computationally very expensive. And what this allows is us to start democratizing science and allowing those who don't have such facilities to actually partake in this. It also allows people to just go to one place to discover data. Now, I'm not going to walk you through the website. The website is at the bottom here and it will also be available later on. But what I wanted to do is, when we talk about these functional analysis pipelines and how we start thinking about putting these together, what we do is we draw on the experience from across EMBL. So there are research groups specifically at EMBL. So I'll point out initially Per Bork and Georg Zeller's groups, where they're all producing tools for the analysis of microbiomes. What we can do is we don't need to invent everything in my group. So we take tools that have been produced by them. So there's one example of the tool we've already included is the mussel tool, where it's trying to understand the microbes there from the functional profile signature. And this tool in itself has actually been built from collaboration beyond EMBL. So using SIB and ETH Zurich, there've also been contributions there. And what we do at EBI, we have other teams that are experts. So for example, the Bateman group produces functional annotations of both proteins and non-coding RNAs, and we bring in those components. And when my team combines all of these to make sure these tools are efficient and comprehensively put together. We have these really large pipelines. I don't want to scare you with this slide. What I wanted to just illustrate from these slides is one of the things that we do is formally describe these pipelines and then make them available such that people can take them and reuse them if they want to extend them or change a tool such for their own use. And it's really important that we have this reproducibility so that people can understand, if you get two different results, is that because the community changed or is it because there was actually an informatics difference? And this is a really important area where we're beginning to find that actually the informatics can cause what we refer to as confounding factors. And what we want is this full provenance of here's the result and this is how we got it using these databases at this time. For those who are not so maybe interested in that sort of informatics part, what we do is this standardized view. So you can start understanding the data sets and we make the data sets all look like one another as much as possible. So in this particular example on the left-hand side, it's a data set that's just become available in Magnify, and it's an upper respiratory microbiome, very pertinent to the current pandemic. And so we can see that this was actually come from the US. And then we have lots and lots of data associated with all the different sequencing runs. But if you want to start accessing that data, you don't have to be an expert. So down the bottom, we have these two buttons. And what we're now doing is trying to allow people to go beyond the Magnify analysis, but simply take slices of the data into things like Jupyter notebooks, where they can then process this. And then they can actually share these workflows in these Jupyter notebooks. So we proliferate this idea of reusing data and reusing people's code and expertise such that we can really understand these communities as a whole. So that's a very brief overview of Magnify. Where I wanted to go next is very much where we're moving to as a resource in terms of as the field evolves, and some of the analysis that we do undertake beyond Magnify. So in this particular slide, there's a very simple illustration of what Magnify does. So we start off with typically short sequence DNA reads. And about 10 years ago, we were purely focused on just analyzing those short reads. But as algorithms have improved and we've got greater access to compute resources, we started to move to actually assembling those short reads into longer contigs. At each step along the way, though, as we assemble those into contigs, we'll then push that data set back into the public archives, so the European Nucleotide Archive, such that other people can reuse those. And so once we have those longer contigs, we can start understanding whole proteins, or even these broader biosynthetic gene clusters that Diane mentioned. But then really where we want to go is not only knowing there's a protein, but where has that protein come from? And this is this notion of clustering contigs we think have come from the same organism. We refer to that process as binning. There may be a bin refinement process where we try and identify contaminating contigs, i.e. contigs that shouldn't have been there. And then we have a set of things that we think are representative of genomes. But because we look at this at scale across many data sets, we can have duplications within those. And so we then try and remove those redundant genomes and choose the very best ones to represent a species. And that's what we're starting to make available in Magnify. So we started to make biome-specific genome catalogs. Within Magnify, when I said this is free to use, the data may be public, already public, and you can ask us to analyze them, or you may have just conducted an experiment, and you can actually upload that data to us in a pre-publication state. And then you can have it analyzed such it remains private. So following that sort of approach, we've applied this to human gut microbiome data sets. Using that, we've applied this to 12,000 of those short read data sets. We then identified nearly 2,000 species. At the same time, there were two other groups doing something similar. And rather than arguing about whose catalogue was better, we actually amalgamated these all together and actually have come up with a fairly comprehensive gut microbiome catalogue of nearly 5,000 species, of which 80% are yet to be cultured. On the right-hand side of this slide, you can see the phylogenetic tree of those species, and coming off the tips of the trees, this green, and those represent those species that are yet to be cultured. And I think this may come up later, and so I won't go into what that exactly means. But what we can understand though, is that in the human gut microbiome, although it's one of the most well studied, there's still huge amounts to learn. But through all those assemblies, so Magnify has now over 28,000 assemblies, we have many, many proteins now. We have 2.4 billion non-redundant proteins. So what can we do with these? So here is one specific example. This was done in collaboration with a company. They have, as part of nylon manufacturing, a precursor is the production of adipole nitrile. And as part of that production, there is a waste stream. But you can transform that waste stream with a nitrulase into something useful. But current enzymes were very, very slow. And so we went mining in that data. And what we could also do, so we went mining in the data, we found 4,000 potential targets. But then we could use the environmental data, such as pH and temperature, to optimize the selection. We had 20 candidates. They were tested. 4 had perfect chemistry and 1 had really conversion rates. This has gone into production as a bioremediation enzyme. Another area where we've used the magnified proteins is through collaboration. So the Holfelder group at University of Cambridge, they've been looking and searching for novel plastic degrading enzymes. So in this top left-hand corner, you've got the petases that are well-known degrading enzymes. And then what they did is they were doing functional metagenomic screens, and they came up with this sequence RR1102. But what they could then do in this sequence similarity network is actually map onto magnified protein sequences and come up with this other sequence, this CI15. And on the right-hand side, you can see an indication of how well these are actually degrading petases. So the best one is already shown in the back in black. And then the one that was discovered in Magnify is the next best one that is out there. Since then, Florian has gone through and actually mutated this and actually now has one of the best performing plastic degrading enzymes out there. And this is how we can start harnessing the microbes. This protein data set has come up in all sorts of different shapes and guises going forward. So many of you have no doubt heard about the revolution of protein prediction from AlphaFold. And so on the left-hand side, we see one of these amazing predictions of protein from the AlphaFold database. So AlphaFold, if you don't know what that is, that's an artificial intelligence system produced by DeepMind to actually try and just determine how a protein folds up in 3-dimensional space from sequence alone. But underneath that algorithm, and I'm not trying to lay claim to anything of the amazing work of the AlphaFold algorithm, But underneath that, the magnified protein database sits. And it is used as one of the target databases. So the way you fold this up is you look for covariance in the alignments to say, okay, these two amino acids are changing. Then and when you see that, they may be close in space. And that's how they do these correlations. And in the paper, it quite clearly said, these metagenomic data sets, they're really useful for actually probing the space that we don't currently know well and do make a significant difference to the accuracy of the algorithm. So just moving to a last example of how Magnify then engages the community. So we've also worked within scientific projects, and I'm just going to illustrate the Hollow Food project to you. So this is about looking at both salmon and chicken production. And the example I'm going to pull on is from the salmon production. And this is about how microbes and the host, in this case, the salmon, interact and how they influence that. And what we wanted to do is start understanding these dynamics. Now, as Ron alluded to, I don't go near a wet lab at all. So collaborators are really important here. So in the middle here, you can see them harvesting gut microbiome data. And these salmon are really important. So they're all in the aquaculture system. And we really need to have different ways of generating protein for actually feeding the world's population. And aquaculture, especially in Europe, is a very key area. And so we want to understand, the influence of the microbes and the salmon in these aquaculture systems. They're called recirculating aquaculture systems, or RASes. And one of the key things that we want to do is look to see how the food influenced the salmon production and the microbiome. So one example of the food additives we tested was actually looking at the introduction of seaweed. Now, seaweed, if you have heard anything about human gut microbiome and people encouraging you to eat things like cabbage or kale, it's really good with the fibrous content and actually promotes microbiome health. The other additive we were looking at is blue mussel. Now, most fish are fed on fish, which sounds really counterintuitive, and we really want to break that cycle. And actually, trying to find alternatives to fish meal is really, really important, and is actually one of the industry has mandated that this must now happen. And so what I wanted to do, and this is really hot off the press because I got this last night, is just to show you one graph about how we've been using this blue mussel protein. So in this graph, what we have along the x-axis is at 0, there's no blue mussel, and then we increase more and more blue mussel into the meal. So 0 actually represents our control. And then each one of these diamonds represents the average weight from a pen. And so what we can do is actually look at the specific growth rate. And the really encouraging thing here is that blue mussel looks like it is actually, can be perfectly substituted for this fish meal. The food conversion rate, so they don't have to eat more of it, is also very, very good to fish meal. There are no changes in the microbiome, which is really encouraging. The only thing we did find out is actually it really depends how you provide this blue mussel. So you can either freeze dry it, which is a bit more expensive than crushing up the mussel with the shell. The fish really don't like the shell. I can understand that. So really, this just really shows you a collection of things we do in Magnify as a whole, and how we are using microbes and how we can actually start understanding how this all fits together in larger ecosystems or in respect to host. In this slide, I'm just illustrating all of the collaborations I have. And I think this is really the take-home message. Nobody knows everything, and you can't do everything. You have to find people who are far better at wet lab than I am to do these things. We can mine the data, but we need people to test. And I really would say that one of the key things throughout my scientific career has been the many fantastic collaborations. And I would just say, finally, thank you for listening. And leave it there, and I'll answer any questions later.